mutation with Dawn Wilgers-Bussell. What are gene mutations, you ask? Well, mutations are changes in a DNA sequence of any organisms. They can also be abnormal changes in DNA. Genes are little pieces of DNA in each of our cells. We inherit genes from both of our parents, making us exactly who we are. Genes are stored in our chromosomes. Chromosomes are strands of DNA that are in some of our cells. Gene mutations happen in our DNA sequences during cell division. They often happen due to an error in our DNA replication, exposure to mutagens like the X-Men, or viral infections. There are two types of mutations. The first is germline mutations. This occurs in the egg and sperm cells. These mutations are usually passed to our offspring. The second is somatic mutations. This occurs in our body cells. These mutations do not pass on. There are many ways genes mutate. I chose the 6 basic ways. First, we have deletion. It involves the removal or loss of one or more nucleotides in the DNA sequence. What is a nucleotide? It is the basic building blocks of our RNA and DNA. A deletion can include the loss of a single nucleotide in an entire chromosome. Deletion mutations is the cause of about 2/3 of cystic fibrosis cases and cat-cry syndrome. Cat-cry syndrome is the partial deletion of chromosome 5. It causes a cat cry in children that have this deletion. Substitution mutation is when one nucleotide is replaced by another nucleotide. Changing one nucleotide can affect how proteins will look and act. This doesn't have much effect on human health and looks, but it can help new treatments for diseases. Duplication is when one or more copies of a DNA sequence, small or large, are made. It happens in all organisms. It's an important mechanism for evolution. Translocation is when a chromosome breaks and then reattaches to a different chromosome. Detection of this mutation can be crucial for the diagnosis and treatment of genetic diseases and disorders. Insertion involves the addition of any amount of nucleotides into a segment of DNA. This happens mostly during the DNA replication process. The effects of this mutation can have no effect at all and can completely disrupt the function of a gene. Inversion happens when a segment breaks off and attaches back to the same chromosome in a reverse pattern. DNA may or may not be lost in the process by small pieces falling off along the edges. This can cause the information to be upside down or backwards, causing a change in the sequence. The human body is just cells working together to make you. Genes have codes that tell cells what to do and what to make. When gene mutations happen on any of the previous slides occur, it can cause a mistake in the code. This mistake can cause an abnormal cell, which could develop into cancer. Inherited genes are given to you by your parents. They're also known as germline mutations. They are the first you. It is in every one of your cells. These will be passed on. Acquired genes are not inherited. These develop at any point in your life. They happen in one cell, then it is passed on to the new cell copied from that cell. These are not passed on. These genes are common cause for cancer, unlike inherited.